lecture, I chose to do Chapter 7, which goes over viruses and prions. And in this PowerPoint, we will kind of go over structures of the virus, the differences between the two, and what exactly a prion is and what it affects. So we'll start off with the differences between a virus and a prion. So a virus is an obligate, parasitic, acellular, small particle that is composed of nucleic acids and proteins. Viruses are composed of either DNA or RNA and are covered in a protein coat. These cause a wide array of diseases in things such as plants, animals, fungi, protists, and bacteria. A prion is actually a small acellular protein which is infectious. These are composed of single proteins. Prions mainly cause brain disease in human and animals, and there's actually no genetic material found in a prion. So we'll start off with the features of a virus. We'll go over its size, its function, and its structure. So when it comes to the size of a virus, they're actually measured in nanometers, and they're typically anywhere from 20 to 200 nanometers. The virus is actually 10 times smaller than a bacteria cell, and it is 100 times smaller than a eukaryotic cell. So it kind of makes you sit back and think, how can something so tiny cause such destruction? And when I talk about destruction, we'll talk about the function of the virus. Its main purpose is actually to deliver its genome into the host to allow expression. It actually forms part of the body's microbiome. An infectious virus is called a virion, and we can actually harness viruses to kind of treat illness, deliver vaccines, and to diagnose infections. So we know that even though they're bad, there's some good to come out of the virus. Last, we have the structure of a virus. Like I stated before, they can either be DNA or RNA. And simple viruses actually have a single molecule of nucleic acid. They're surrounded by a protein coat, and we call this the capsid. But there is also another type of virus that is not surrounded by that protein coat, and we'll go over that next. But viruses also do not contain nuclei. So as I stated, we'd go over this. These are called enveloped and non-enveloped viruses. So we'll kind of go over the differences of these, and we'll start with an enveloped virus. These actually consist of a lipid bilayer that closely surrounds a shell of a virus-encoded membrane-associated proteins. It also contains a viral genome and core proteins that are wrapped within one or more membranes. Non-enveloped does not contain an envelope. Hence, that's where the name comes from. We also sometimes call these a naked virus. This contains nucleic acid and a protein coat, a capsid, and it usually enters cells via endocytosis. So here I provided a picture so you could visually see the difference between an enveloped virus and a naked virus. And you can see with the enveloped virus, it shows you where that nucleic acid is, where the capsid is, the envelope, and the spikes. The only difference from the left to the right is that envelope. The non-enveloped also has that nucleic acid, that capsid, and they have spikes. But you can see the envelope, which is this yellow rim around the virus to the left. So when it comes to giving viruses names, they go by this binomial nomenclature. With this, they are split into family and genus. The families are named with a suffix viridae, while the subfamilies have the suffix virinae. Genera, the suffix is virus. So the prefix may be another Latin word or signa. Abbreviation, which is an abbreviation derived from some initial letters. So here I kind of put the binomial nomenclature hierarchy. Domain is at the top, and then you will go to kingdom. Next is phylum. Then we have class, order, family, genus, and species is right there on the bottom. And some charts will actually have subspecies listed below that, but species is on the bottom. Next, we will talk about the multiplication cycle of an animal virus, and there are 6 steps to this. So we have attachment, penetration, uncoding, gene expression or replication, assembly, and last, release. We'll go in depth on this in a couple of slides. So our first step is called attachment. This is when that virus attaches or binds to the host cell, and then interactions between the viral proteins and the molecules on the surface of the host cell begin to happen. And this is the first step and must happen for this process to start. Next, we have penetration, and this is actually the second step. It occurs through contraction of the tail sheath of the virus. When this happens, it will inject its viral genome through the host cell's wall and membrane, but the phage head and remaining components will stay outside the cell. They do not enter the cell. Next, we have uncoding, and this is the third step. So this is when that virus will break down or remove the capsid that we talked about earlier, that kind of protein layer. And once that happens, it causes the release of the viral genome, where replication and transcription will take place. On to our fourth step. This is the gene expression or replication process. And as you can see, there are 3 routes. Route 1 is when the transcription of dsDNA occurs. Route 2 is the transcription of ssDNA. And route 3 is the transcription of dsRNA. For our 5th step, we have assembly. This is where all those virus particles will come together at one site in the cell. Once they're together, new viral particles are assembled from the genome copies and viral proteins using the host cell ribosome. And for our 6th and final step, we have release. And this does exactly what it says. The virions are released from the cell. So this is where they can go out and start replication and cause disease. And some viruses actually exit the cell by means of lysis, meaning they completely burst the host cell. Now this just completely destroys the cell. There's no coming back from lysis. Next, we'll kind of talk about cytopathic effects on cells. So this is when structural changes in host cells are caused by viral invasion. The infecting virus causes lysis of the host cell, which we talked about before was that bursting of that cell, and there's no coming back from lysis. Once it's gone, it's gone. And it can also cause changes in the cell's morphology. The shape, an example would be like rounding of a shell. It can cause the shape of the cell to completely change. And then we have the cultivating of viruses. So viruses can actually be cultivating within a suitable host, such as a living cell. The purpose of this process is to isolate and identify viruses in clinical samples. So we can identify different viruses by isolating them to see their structure and things. And like I said, this is to do research on that viral structure, replication, genetics, and even the effects that this virus would have on a host cell. So, 3 types of tissue culture are organ culture, an explant culture, and a cell culture. And here we'll kind of talk about prions and the brain. So, disease actually occurs when normal prion protein becomes an abnormal clump in the brain. This then actually causes brain damage. Um, it is an abnormal accumulation of protein in the brain. And we know that our brain is very important and we need to protect it at all costs. This can actually cause memory impairment, personality changes, and difficulty with movement. So, prions in the brain are a big no-no. Prion disease is actually really rare. It is progressive and it is fatal. Currently, it is an untreatable degenerative disorder of the brain. So prions cause brain damage, and that is the end.